Here are a few examples of cells that are differentiating differently based on the signals they're receiving from the environment. So muscle cells can be found in many different ways. You can have cardiac muscle, skeletal muscle, or smooth muscle cells. Cardiac muscle and skeletal muscles both have striations, but one of them has multinucleated cells, while cardiac muscle has nuclei that are more sparse within the cells. They are all designed to contract and relax, allowing for their movement. However, depending upon the function they need to perform and where they are in the body, they obviously look very, very different. So even though both skeletal muscle cells as well as cardiac muscle cells are able to contract and relax in a rhythmic manner, cardiac muscle cells do so rhythmically all together, coherently in the entire heart, while skeletal muscle cells will do so in a more bundled up type of fashion. So some of them might be moving while others are stationary. Another example of differentiated cells from non-differentiated cells to begin with are blood cells. Blood cells are all going to be differentiated from the stem cells found in bone marrow, and those are going to be changing into various types of blood cells depending upon the needs of the body. So you can have your erythrocytes, which are red blood cells. You can have various types of white blood cells, including your neutrophils, basophils, monocytes, and then you can also have your platelets. Each one of them have very different functions and play a very different role in the body as part of either the defense system or the transportation system. Finally, something that we are going to be working on in this lab exercise is the nerve cells. So you can have neuroblasts in your brain. Even in adults, there are small quantities of neuroblasts that can be found in the brain. These stem cells can be changed only into neurons or functional nerve cells. However, they are relatively hard to differentiate as we age compared to other systems where their progenitor or precursor cells are more easily transformed and more easily differentiated into the adult types. But these neuroblasts can be differentiated into nerve cells given the right environment and the right signals from the environment. The nerve cells are designed to receive and transmit impulses from one area to another, so they require not just the cell body to be differentiated, but to have these large and small projections that come out of the actual cell body within, and those are what transmit the signals. You have your dendrites and your axons that can then transmit the signal from one cell to another or from one body part to another. Now, inside the body, the nerves, the actual functional nerve cells require Schwann cells, which are helper cells that allow for the signal to be transmitted quickly from one part of the nerve to the other. However, when we culture them in vitro, we are not able to necessarily create them all the way through with the Schwann cells the way they would inside the body. Still, we can examine the process of differentiation from a neuroblast to a nerve-like cell in culture by using the right differentiation factors. So two of the experiments that we've done at various times in this lab, one of them is using mouse neuroblast cells. These are N2A cells that are isolated from mice. These are neuroblasts, so they are not completely totipotent, but they partially differentiated stem cells. They can only be changed into one of the nerve cells inside the body or outside in culture. Normally when you look at them, you'll see them in these tiny little balls, very small, kind of clustered together. They almost look like a bunch of grapes or bacterial particles, except these are larger than a typical bacteria, obviously. But these cells are going to be clustered up into these little clusters. They are rounded. they don't attach very well to the surface of the plate, and they grow in this manner. As they start to differentiate, they will start to flatten out, and this is an example of those early differentiation points. So you have some of these cells out here that are early differentiated cells back here as well. So these cells are beginning to flatten out, so you can see a flat cell body with the nucleus inside, and they are not so 3D in shape and not so rounded up in shape as they were when they were a neuroblast. Then as they go further along the differentiation, you start to see projections coming out of them, one or more, and these projections are what would transfer the signal from one cell to another and from one part of the body to another inside the body. And finally, as they differentiate to their final nerve-like state, you will have long projections that are connecting to each other, that are communicating with different cells. So there are projections that are interacting with projections from other cells, and several smaller ones that can also be used to transmit signal. So here is an example of the experiment that you will be doing. Normally, we keep the cells in really high serum to mimic the environment that the cell would be in as a neuroblast, as a stem cell during embryogenesis. This high serum maintains it in this undifferentiated state. If you culture them long enough, they automatically start to become a little differentiated, but overall you'll still see several cells that will be still state 0 or very minimally differentiated cells. If you remove the serum and kind of mimic the environment of the brain, where you would be expected to see a lot less serum in the system, because you have the CSF, which does not contain as much albumin and growth factors inside, they start to differentiate already, and you start to see some early differentiation states. Sometimes they even differentiate to stage 2 or even stage 3 neurons, but many of them will still be partially undifferentiated. Finally, if you were to use a specific agent, specific molecule that is known for differentiation, like retinoic acid, you can see the more advanced differentiation state of these N2A cells into neurons. Another example that we've done in the past is with, again, myoblast cells from mice. These are C2C12 cells that can be used to create myotubes in culture when given the right differentiation factor. So these cells, as myoblasts, are extremely small. They are clustered up. They look very similar to our typical cells that we see in culture, like what you saw last week with MDA468s. But as they develop into myotubes, they become multinucleated, and here is an example of them, and they become elongated into these myotube-like structures. If you culture them well enough, you would even see them contracting and relaxing in small clusters within the plate. So whichever differentiation agent we are using, many times it needs to be dissolved in some type of a solvent. The solvent that we are going to be using for retinoic acid is DMSO. DMSO is a great carrier molecule, so things that may not be so soluble in water may still be very soluble in DMSO. It dissolves a lot of stuff, and it carries a lot of molecules. It is capable of carrying a lot of molecules like growth factors and small molecules that you would find in the extracellular space that can help the cell grow and divide and differentiate. So retinoic acid, when combined with DMSO, is going to have this complex where retinoic acid is bound to DMSO. DMSO can then carry it through the cell membrane, through the lipid bilayer, into the cell, where retinoic acid will dissociate from DMSO and bind instead to a retinoic acid receptor. The retinoic acid receptor translocates into the nucleus, where it binds to specific rare elements. That stands for retinoic acid response elements. And these elements, wherever they are, whichever gene they are on, are going to then get activated, and those genes will get activated in return and transcribe the genes and proteins responsible for differentiation.